Hello everyone, my name is Srilega VM. In this video, we are going to discuss about 454 sequencing and illumina sequencing. 454 sequencing. 454 sequencing can sequence fragments of DNA. The DNA is broken up into fragments of around 400 to 600 base pairs using restriction enzymes. Genetic adapters are added to the ends and these are annealed to beads 1 DNA fragments per bead. Short sequence of DNA called adapters. DNA sequence on the beads are complementary to sequence on the adapters. When the DNA fragments attached to the DNA on the beads, the bonds joining the double strand together break and the strands separate, becoming single-stranded DNA. The fragments are then amplified by PCR. This creates millions of identical copies of DNA sequence. The beads are then filtered to remove any that have either failed to attach to any DNA or contain more than one type of DNA fragments. Each bead is then placed in a single well of a slide. The well contains DNA polymerase, nucleotide and primer needed for the sequencing. The polymerase enzyme and primer attached to the DNA fragments on the beads. When base is incorporated into the DNA, light is given out and this is recorded by a camera. The addition of each nucleotide releases a light signal. These locations of signals are detected and used to determine which beads the nucleotide are added to. This kind of sequencing generates graph for each sequence read. Illumina method. Illumina sequencing has been used to sequence many genomes and has enabled the comparison of DNA sequence to improve understanding of health and disease. Break up the DNA into more fragments of around 200 to 600 base pairs. Short sequence of DNA called adapters are attached to the DNA fragments, then made single-stranded. This is done by incubating the fragments with sodium hydroxide. The complementary DNA bind to primers on the surface of the flow cell. The DNA attached to the flow cell is then replicated to form small clusters of DNA with same sequence. Nucleotide bases and the DNA polymerase are then added and joining the strand of DNA attached to the flow cell. This creates bridges of double-stranded DNA between the primer on the flow cell surface. The double-stranded DNA is then broken down into single-stranded DNA using heat. Addition of primer and certain nucleotide, they are fluorescently tagged. The DNA polymerase then bind to the primer and add first fluorescently labeled terminator to the new DNA strand. Once a base has been added, more bases can be added to the strand of DNA until the terminator base is cut from the DNA. Lasers are passed over the flow cell to activate the fluorescent label on the nucleotide base. This fluorescence is detected by a camera and recorded on a computer. Each of the terminator bases give a different color. The fluorescently labeled terminator group is then removed from the first base and the next fluorescently labeled terminator base can be added alongside. And so the process continues until millions of clusters have been sequenced. Thank you.